The overall goal of the following experiment is to assess the effects of various molecular and pharmacological manipulations on dendritic spine morphology and motility. This is achieved by transfecting primary cultured cortical neurons with GFP with or without exogenous expression of DNA plasmids to outline neuronal morphology and manipulate endogenous protein expression and function. In the second step, cells can be treated pharmacologically and subsequently fixed and immunostained, or prepared for time-lapse imaging and then pharmacological treatment, which will allow for the visualization of dendritic spines and the induced changes in dendritic spine morphology and motility. Next, Z-series images of fixed cells or images of live cells taken at specific time points are acquired using a confocal microscope. Detailed measurements of dendritic spine morphology can be made from 2D projections of Z-series images. The results obtained show the effect of molecular or pharmacological manipulations on dendritic spine morphology or motility based on quantitative morphological analysis of dendritic spine shape or motility.
This method can help answer key questions in neuroscience, such as how is synaptic plasticity regulated, how do excitatory synapses develop, how are synapses maintained or eliminated, how molecules such as neurotransmitters or genes regulate synapse structure.
Though this method can provide insight into the basic mechanisms that underlie the regulation of synapses, it can also be applied to investigate the contribution of disease-associated genes to alter synapse structure, which is a hallmark of a number of psychiatric and neurological disorders, such as autism, schizophrenia, or Alzheimer's disease.